We are very lucky to have this week our very own Professor Anne Stone. Many of you know Anne, and if you don't, you certainly should know Anne. She's a really phenomenal colleague and an amazing scientist and researcher. So Anne has come a long way and ended up here at Arizona State University. First, she did her bachelor's at the University of Virginia, and then went on to do a master's and a PhD at Penn State in anthropology. After which she went on and did some postdocs and moved around a little bit at Penn State and at the University of Arizona. Spent some time in Germany. That was probably before then, actually.
That was before that.
That was
Yep.
before, right, at the Max Planck. And then was started as a first time as a professor at the University of Mexico, just across the border, one of these directions. And we were lucky enough to bring her here. This is before my time, but lucky enough to bring her here to Arizona State back in 2003 when she came in as an associate professor, and then quickly rose through the ranks and is now a Regents professor, which is kind of a big deal here at Arizona State, and has won a number of awards. And if I were to go through all of them and talk about all of her publications and work, she would not get to talk today. But some highlights here is that she was elected to the American Association for the Advancement of Sciences back in 2011 and to the National Academy of Sciences here in the US in 2016. And excuse me, must be that smoke. And most recently, she was awarded a Guggenheim Fellowship, which is ongoing still, I imagine, or that was a...
I think technically it just finished.
Okay, technically it just, all right, so she's no longer a Guggenheim Fellow, but that's okay. So, Dr. Stone's studies has been a leader in the field of ancient DNA for decades, developing and deploying tools to extract, sequence, and draw meaningful biological insights from very old and not very well preserved DNA samples. She pairs these data with sequences from modern human populations to understand how disease and diet have shaped human evolution. From being the first to sequence Neanderthal DNA, to identifying how genetic variation in the salivary amylase gene was likely selected for in some human populations so they could eat their starchy diets. Her work has three main foci. One is Native American population history. The other is the evolutionary biology of the Great Apes. And the last one is understanding the evolutionary history of pathogens in humans and other animals, which she will tell us a little bit about today. And if this didn't already impress you, as an amateur baker, I am envious of Anne's ability to make what is probably 1,000 different types of cookies. And hopefully I will approach that at some point in my career as well. With that, let's welcome Dr. Stone.
Christmas baking. Thank you. It's a pleasure to be here. I was telling people earlier, my voice is a little gravelly because of the smoke, which triggers asthma in me. So hopefully this will go okay. I should first give you a warning. There are a few pictures of human skeletal elements and remains in this presentation. Secondly, I'm going to kind of give an overview about what we can learn using ancient DNA about pathogens, and I'm going to focus really on two projects, areas of research in my own lab over the course of it as well. So before the COVID pandemic, I used to really have to explain this more, but I feel like people understand it a little better now. You know, and obviously I'm preaching to the choir here at SEM. Why is the evolutionary history of pathogens important? But I always like to make this point, you know, just if we look at human history, even recorded history, but also further in the archaeological record, there's a lot of indication that these have had a major impact on human history. This is from the 1918 flu pandemic. We can also tell from genome analyses that they're really the greatest selective force acting on our genome. It's always immune loci that have some of the highest selective peaks when we do these kinds of scans. And third, you know, really understanding how these pathogens work, hopefully, ideally, also helps us design better drugs and vaccines in the future. You know, we need to know our enemy, so to speak. Now, a little bit about ancient DNA. And also, this slide is sort of randomly in here. Okay. Oh, I know what I did. All right. Hopefully these are the right slides. If not, this presentation will veer off into another direction. Yeah, that'll be good. Okay, ancient DNA. I'm going to explain how we do a lot of the ancient DNA research here in a minute, but in the early days, some of the early attempts were really trying to, we're pretty limited because of PCR. These were in the early days. And there were a lot of questions people wanted to ask, but some of the really simple ones were, does this individual from this burial context have the disease we think they have, if there's diagnostic markers or not? And is there some way to figure that out? And it was really challenging. So this 1994 paper, basically what they did was PCR, a repeat element found in 0 to 27 copies in the Mycobacterium TB complex. This is a 1,000-year-old Peruvian mummy that had what looked like from paleopathological analyses to be a case of TB with TB-infected lymph nodes. And ta-da, they got a band. Right? It amplified, and they sequenced it, and it was, in fact, this repeat element. But, this is kind of the prehistoric times of ancient DNA, right? So those of you who ever did a Sanger sequencing with the joy of radioactivity will recognize this. And this was done at about this stage of floppy disk ness. So I like to point this out because it was really challenging, both from a technical perspective and also from, the analyses that you could do. And also, I should point out in terms of comparative data, we did not actually even have a phylogeny for the MTB complex at the time, other than some very basic ones that showed us things we already knew. Guess what? Mycobacterium tuberculosis is related to Mycobacterium bovis. You don't say. So, there were real limitations in what you could do, both technically and in terms of analyses. Thankfully, times have changed, but we still have some real challenges. And this is kind of a summary of those. We still have the issues with ancient DNA of the low quantity and quality of DNA or RNA in the case of many viruses. We still have the challenge of a lot of bacteria and viruses are not known to science. That has become less of an issue, which is useful, but it's still, in fact, an issue. We have to think about the weird ways that pathogens evolve. They often have these boom-bust population dynamics. It makes looking at signals of selection tricky. It makes sometimes following transmission patterns across the landscape tricky. It certainly can make mutation rate estimation tricky. We also have issues in many pathogens where recombination and lateral gene flow can make the phylogeny more of a weird reticulation that's nasty. Plasmids can be transferred. All these sorts of things can affect or can make life a bit challenging. When you come specifically to ancient pathogens, we have to think about the possibilities for contamination, not just from the laboratory environment, and I'll show you some things that we do in the lab to prevent this, but also contamination by environmental bacteria or viruses that may be closely related, okay? I think the other thing that I always remind, particularly archaeologists, is, just because we find a pathogen doesn't mean it's what caused their death. They had it, but many people in the past were co-infected. This is true today. It was certainly true in the past. So we have to be cognizant of that. I think the other real challenge is most of the pathogens we've looked at, and now there are quite a few, it's quite exciting, using ancient DNA methodologies are known pathogens. There's real questions about whether we could analyze an extinct pathogen. And there are some maybe out there. So one would be like the sweating sickness that's reported in Tudor England. We have no idea what that was. And, you know, I'm not sure if we ever will, but so that's a real challenge. The other issue is pathogen load. You have to think about where in the body a pathogen is found and how much of it is there. There's a lot of, well, there's a number of pathogens that do leave characteristic skeletal indicators. Those are chronic things like TB, syphilis. But by the time somebody shows skeletal indicators for syphilis, there's usually almost no bacterial load. A lot of that is a reaction, an immune reaction, right? So pulling syphilis, the treponemal bacteria out has been a real challenge. The other thing is if you die really fast from smallpox, Ebola, pick your pathogen, plague, your bones will look great, right? You died too fast for there to be any mark. And so, you know, that's hard to pick out. It's hard to know where to sample. And we also have real biases in the archaeological record. Sometimes people made cemeteries and they buried people in a primary inhumation. Sometimes they used cremation. That's not good for DNA. Okay. And also, you know, we see a bias just in terms of the ancient DNA data over space. And I'm going to go into some of these here in a minute really quickly. But which I probably should have done rather than stay on that slide, but there we are. How do you analyze ancient pathogen DNA? And here is one of my graduate students in the ancient lab. That's not you. I think that's oh no, I think that's Felicia. Okay, so anyway, right. So you take a sample, whether it's from a skeletal element, from a piece of tissue, from even a formalin-fixed paraffin-embedded slide that's from an archive, and you extract the DNA, and typically less than 1% of the DNA in the sample is actually endogenous. And even smaller amount is the pathogen you're interested in. Next up, we make a DNA library, and we do this because, you know, we are extracting all the DNA, even from the burial environment, and we often need to search through that, and a library helps us immortalize things. So we have the adapters, indices, lets us pool samples, because these are basically barcodes that differentiate samples once they're pooled, more adapters that basically let us manhandle things. We then typically we'll often do shotgun sequencing just to see what's there and run it through things like Kraken or Malt that can analyze that type of data and kind of tell us give us a hint to what's there. But usually we then have to move to capture. And what we do there is heat up the library to make it single-stranded. We have biotinylated, or we have baits. Those get attached to beads, biotinylated, magnetic, and this lets us pull the DNA that we're interested in to the side of the tube and move the rest of it elsewhere. Okay, so this is how we enrich for the DNA of interest. In our case, it's pathogens. We then take that, sequence it, and do various analyses. One other point about ancient DNA is that it is typically shredded. Over time, hydrolysis and other degradative processes basically result in the shredding of the DNA into fragments. And unfortunately, most of the extraction methods that you're used to lose most of these small fragments, right? So this peak here in blue is from shotgun analysis. This is mitochondrial capture as an example. And what you're noticing are the peak sizes are around the 30 to 40 base pair range. In the mitochondrial capture, you'll notice this steep line. This we run into because most bioinformatic methods can't align sequences that are shorter than 25 to 30 base pairs. So that's kind of our cutoff there. So there's DNA in that size range. We just can't really place it well. The other issue I mentioned was DNA damage. In the process of the DNA being shredded, you get these overhangs that are single-stranded. They are prone to deamination of cytosines. And as a result, we get what look like C to T transitions that are actually because the polymerase sees that deaminated cytosine as a uracil. And so bioinformatically, we scan the sequences and remove these ends to make sure that it doesn't bias our results and make mutation rates look really big. Okay, so I wanted to touch base really quickly on a couple of these pitfalls and challenges and then get into what we can do. So I've already mentioned these issues. One other thing I will add, though, is this is a particular problem, contamination by close environmental relatives for Mycobacteria, particularly Mycobacteria tuberculosis, MTB, Mycobacterium tuberculosis. Mycobacteria in general, most of them live in the environment, and mostly they are found in soil and water. And that is mostly where people are buried. And so we end up, when we study tuberculosis, really trying to separate the wheat from the chaff, as it were. And you can see that here. This is an analysis where we were trying different methods of pulling apart environmental from the actual pathogen. And all of this turned out to be environmental mycobacteria. Okay. I mentioned also these just general challenges that can occur depending on the pathogen of interest. You know, this of course is from Next Strain, and this is regarding COVID, but you know, certainly we could see in the analyses of the COVID virus during the COVID pandemic, the analyses of SARS-CoV-2 really showed some of these boom bust patterns that can affect estimates of rate for the molecular clock. So we do have to be mindful of these sorts of things over time. Pathogen load, as I mentioned, is an issue. What we find with tuberculosis is that most of the time we have to sample really closely to a lesion. So you can see the remodeling that has occurred. You can see where the vertebral bodies look a bit thin in spots. These are TB lesions. Kelly Blevins, one of my former graduate students, did a really nice study sampling different locations to identify, you know, what was our best bet for recovering Mycobacterium tuberculosis complex strains, DNA from individuals who died with TB. A lot of pathogens, if there's a septicemia or in the case of leprosy, the pathogen load can be higher through the blood. In the case of leprosy, because leprosy is sort of concentrated, you know, one of its areas of higher load is in the face, nose and mouth. Tooth pulps can be a really useful place to isolate the DNA, and that tooth structure helps protect the DNA from environmental degradation. So those are, as I mentioned, issues that we often face. And I just bring up those two pathogens because I'm going to talk about them more. I've already mentioned the issue of preservation. Ancient DNA is preserved better in cold, dry places. And also, the funding is better in Europe. So we've got great coverage. Those really good EU grants. We've really got some really wonderful ancient DNA studies in Europe. And our studies of microorganisms are clustered there. It's really hard to see that on this map. And then there's a few dots in the Americas, mostly because of our work, actually. And so the other place where we see more work done is looking at the microbiome in dental calculus. And dental calculus does seem to help preserve some of that record. Okay, so I've talked about all the problems. I should talk about some of the promise here, and why this research is so exciting and why we still do it is because when we can get results, it's great. And we can find out about pathogen origins. We can look at the relationship of lineages across space and time. We can try to say something about maybe when a pathogen actually jumped into humans and look at that process of adaptation. We can also turn that around and look at the hosts and see maybe, were there certain people who were more susceptible to that pathogen compared to others? And there is beginning to be data about that in particular. So the first, and then also you can combine this with other archaeological information to look at how human culture has affected disease risk. All right, so the first one I want to talk about is Hansen's disease or leprosy. And this, we think, transmission is via the respiratory route. Mycobacterium leprae is related to TB, but distantly. And it causes lesions in the face. It also causes numbness in the extremities. And as a result, people will often damage, particularly fingers and toes, and not realize it. And they'll get a secondary infection and gangrene, and this can result in loss of fingers and toes. So it doesn't directly affect, cause this loss, but it contributes significantly. M. lepromatosis is newly discovered, but is also newly discovered as in the last, 5 or 10 years. But it's also a causative agent of leprosy. We haven't found that yet in ancient DNA, but it's today a neglected tropical disorder, and has in the skeletal record diagnostic changes. Now, in my lab, one of the things we're doing is using formalin-fixed paraffin-embedded biopsies to look at the distribution of strains in the Pacific and the history of infection there. We're also interested, well, I'll get into the other questions, but one of the things that you see in modern patients, and this was true in the past as well, is a range of a classification range about how people are affected. This is from a historical Scandinavian medical text. And you can see people with the milder, what's known as paucibacillary or tuberculoid leprosy, and people with the more severe form, lepromatous or multibacillary. This is really probably largely due to how your immune system fights off M. leprae. People with this their immune system's doing a better job. Here it's doing less. People can move between these two. Once, though, you have tissue damage, it doesn't get undone. One challenge, though, is obviously there's more pathogen load out here compared to here. And so even in modern patients, it's very difficult to even look at the DNA, the genomic DNA from M. lepra. So most of our samples have been successful in this group here. We started analyzing biopsy and also slices that were mounted on slides from primates. And one of my former graduate student, Thunby Honop, looked at macaques, chimpanzees and pseudomangabes with leprosy. And interestingly, and you can see that the facial changes are similar, also some of the skeletal changes. But interestingly, these cluster with human strains in the same region. So we don't have a really good sense of where M. lepra hangs out or how it jumped into humans. We do know based on its genome, it has a really downsized genome that's kind of characteristic of an obligate pathogen. It's likely been a pathogen for millions of years. So it hasn't just been in us. You'll also notice, though, that we gave it to armadillos. I'm sure they were pleased. And then they also weirdly found it in red squirrels in the UK. So we don't really have a good sense of the distribution of animals affected. And one of my graduate students, Adele Crane, is looking at some of that right now. The other thing that's really interesting is people assumed that M. lepra was a really old pathogen, but once the ancient data from other groups came into play, and we could use that to help calibrate the molecular clock, we realized that the coalescent most recent common ancestor is about 3,500 years to 4,000 years. So at least for all the strains we've looked at to date, and even with newer data, these dates hold. So what was it in prior to that? We don't know. So that's a big question there. And our, that's just getting into some extra info. All right, so in the Pacific, what we're interested in is the diversity of strains there. And part of the reason we are interested, there is a skeletal record of M. lepra. It's pretty sparse in the Pacific. Austronesian expansion began about 5,000 years ago, but then really gets out into remote Oceania by about 800 to 1,200 years ago. And they had some really amazing trade routes. And we also, just because of their the Polynesian amazing voyaging abilities. But at colonization, at contact and afterwards, there were different patterns of colonization in the Pacific, and most of the samples that I will talk about are from the US states and territories in the Pacific, but we're working to expand that. The other thing, though, is that there were not just contacts from Americans, but also from Asia, from China and Japan, with trade in that direction, and then not just Americans, but British and French as well. So we're interested also in what might have happened after contact in terms of moving strains or introducing new strains. Now, leprosy from the oral tradition, or Hansen's disease from Hawaiian oral tradition, is known as the disease that separates families. It also can be called the Chinese sickness. And on the island of Molokai, a leper colony at Kalapapa was established in the 1800s and still in use, although people had been treated prior to that, but still in use until 1969. So we sampled, this is in collaboration with Christopher Lum and others, but we sampled these paraffin-embedded biopsies from sites around the Pacific in our initial sampling. And what we found was that I should back up and say there were a few published strains that you can see there. The ones in red are the ones we added in. But what's really interesting is that these are the two, branch 0 and branch 5 are the two most basal lineages within the phylogeny. Okay, so this is one of the reasons why I thought the Pacific would be a really interesting place to look. to find out more about these really deep lineages and their distribution. Our analyses didn't really change the time to the most recent common ancestor. Now, since then, we've added additional samples. Let's see, we can fly them in. And actually, I don't have enough circles because we now, as of, I think, a week or two, or a few weeks ago, have 12 new genomes to add to the mix. They range from 1x to 25x, and they all still fall in branch 5 and branch 0, so they're still part of that. In our previous analyses and in our preliminary analyses so far of the new data, what we see is what looks like a pattern consistent with the initial migration into, oh, I have more dots. Okay, more dots. Okay. We have a pattern consistent with the spread of M. leprae actually with the first, either with the first colonists into Polynesia, so not European or China, Asian colonization later, but that early occupation of the region, or spread via trade routes. And that's something we'd like to look at further. So just to kind of give you a sense of our future plans, we are now trying to expand the sampling into other parts, French speaking, for example, and other territories and states in Polynesia. I may have to go tour the South Pacific. It'll be, I know, I'd have to wear SPF 800. And hopefully, so far our data are from blocks that range from the early 90s to about 2015. But we are hoping to get older archival samples. There are, for example, some slides from the 1940s from Hawaii that we hope to analyze. So we hope to expand that and look really at the pattern of variation in the Pacific, whether it supports that introduction during initial colonization, or if patterns of trade later brought it in. How common are antibiotic resistance alleles? In a lot of, drugs to fight leprosy become available in the 50s and 60s, but they're not free from WHO until about 1982. So when people started being treated varies. So we may be able to look at pre-drug treatment and post-drug treatment examples to look at patterns of selection there. And we'd also like to look, often there are multiple biopsies from the same individual, so we can look at how much within patient diversity is there. Now, the picture of, I mentioned that our samples have fallen into these basal strains. What's really weird is all the rest of the ancient, really ancient data is from basically Europe and one from Egypt. And interestingly, 0 and 5, well, 0, not 5, but branch 0 strains are also found there. Not commonly, the most common is 3, which is in green. So it's really kind of unclear when and where. Well, the when is a little clearer, but the where is not clear in terms of when M. leprae jumped into humans. But we do, this is an analysis of population expansion of the pathogen through time, its increase over time. So this is really a case where the One Health approach is important. Ancient DNA adds information in terms of calibration, in terms of the distribution of strains over time. And then the One Health is really teaching us that we've got to look more broadly. And recently, my graduate student, Adele Crane, has gotten a M. leprae strain from a panther from South America. Maybe it just ate an armadillo, we don't know, but it's actually from the spleen. So so that's another area of interest, but it's really hard to find. Very quickly, ancient DNA has done a lot. If we, and this is not my work, but if we look at the example of the plague, this really confirmed that plague, which is known for these historical first Justinian plague and the Black Death, and then more recently the third pandemic. This, of course, we knew was caused by Yersinia pestis. These we thought were, but really ancient DNA is what confirmed that and showed how these were all related, that they do come out of probably Central Asian plague reservoirs. But what's more fascinating, I think, in many ways, is that because it's found in dental pulp during the huge pathogen load of the illness, these folks, you can pretty well guess that that is in fact what they died of. But surprisingly, studies of that were trying to look at human DNA and human population history found really old Yersinia pestis from the late Neolithic and the Bronze Age. And what's really fascinating about these is that you can see the changes in the pathogen over time that seem to make it easier for transmission via fleas, and perhaps tell us something about differences in plague over time. There's some debate about whether or not it could cause bubonic plague, for example, that form early on. Lastly, I want to talk briefly about TB, which is another area of very active research in the lab. The thing I want to remind you first, though, is that the disease we call tuberculosis is actually caused by any of these things. Which one these were, if they did some testing, they respond to the same drugs unless they're drug resistant, and then you've got to start a different set. But this complex species, subspecies, ecotypes, they're kind of ecotypes really, all cause TB. And it's kind of described as a wasting disease. People with good immune systems can wall it off in a granuloma. But if you get co-infected by something else, so for example, people with HIV, the #1 cause of death for people with HIV is TB. It's not HIV. And that's because TB loves to co-infect, and it's a wasting disease. And then it can be pulmonary, but it can also affect other parts of the body. And if it's chronic, it can affect the bones. And this is an example of chronic impact of TB on the vertebral column. It causes basically the vertebral bodies to collapse and the spine to kyphose, and this is known as Pott's disease. So when we think about TB in the past, we have to ask, okay, what was it? When we see individuals in the record with these bony signatures of TB. Initially, people thought that we got TB from cows during the process of domestication. However, archaeological finds in the Americas clearly showed TB of some sort was present pre-contact. And then in the late 90s, early 2000s, when people actually got around to building a phylogeny, they noticed that M. bovis was much more derived than the human M. tuberculosis strains. So that also pointed to cows not being the issue, and possibly we gave it to cows. So people then also started sequencing first genes from the different strains and then complete genomes. We now have over 10,000 complete genomes from around the world. And the general pattern that you see here holds up. We have these European American strains in red. We have the Beijing strains in blue, these purple. These are the ones that spread, are most common today. These are deeper lineages, and you'll notice the animal strains are tucked into the African strains, human strains. So this is the picture today, but what was the picture in the past and what was the picture in the Americas? And so in 2014, we published a paper where we looked at individuals buried in the Osmore River drainage. These are 3 sites that are about 1,000 years. Pretty decent coverage genomes, and we found a couple of surprising things. So the first surprising thing that we found is that if we used the archaeological dates to calibrate the phylogeny, because there was a huge debate about mutation rates in TB, we get a most recent common ancestor that's somewhere in the 3,000 to 5,500 year range. Much more recent than we thought. The other thing is that, oh, see, I did it out of order. Right, there we are. The other thing is that the ancient Peruvian humans had TB strains that were most closely related to those seen today in seals and sea lions. And that was a huge surprise. We actually had to spend some quality time finding pinniped strains to make sure that this was correct, because the one strain, the one genome available was from a zoo context. Now, if we look at the archaeological record, what we see in the Americas is that South America has the first documented cases. So this actually fits our findings, because originally we had thought people had brought it actually during the initial migration into the Americas, 15,000 years ago, maybe they brought it. And then you would expect the early cases to be in the north, but they're not. We see cases later in North America, actually. So it turns out to be because of this. However, we got some critiques of our work. But before I get into that, people said, oh, well, maybe this is just zoonotic and all the other stuff was caused by other strains. The thing to point out about the M. pinnipedi is that I got to learn a lot about seals and sea lions, and it's only these guys that have the Southern Hemisphere strains, seals and sea lions that have TB. Northern Hemisphere seals and sea lions do not. But we had a bunch of questions. Were there multiple jumps? Do we see readaptation? Does it spread via human to human contact north, or is this because of other contacts with the Americas before European contact, or at least before the Vikings? So we also looked at sites up 2,000 meters that are under present-day Bogota, Colombia, an area noticeably lacking in seals, or close proximity to the coast. So here are the other sites. So these guys certainly were fishing or going to the coast often. These guys, not so much. We also got a genome out of even further away. What we found in this case was, again, that here are the modern pinniped strains. Here are the ancient human TB strains from South America, and they all cluster together and are most closely related to these modern strains. Now, these branch lengths are shorter because this is 1,000 years earlier than today. Okay. We also have new data from Tenochtitlan and Tlatelolco, analyzed by and in the process of working on manuscripts by my former graduate student, Kelly Blevins. Okay, need to wrap things up. All right. So she's very happy because she got good data. Tenochtitlan obviously was the Aztec capital. Tlatelolco was the adjacent and basically became part of the city, but it's basically the marketplace. And so the question is, how does the TB found here relate to the TB found in South America? And we also retrieved a new genome from northern Chile. So just to zoom on in, the Aztec capital is located, or was located, basically under what is now Mexico City. This lake is gone. It was drained pretty much. There's a few remnants, swamps, but the context that we're analyzing is from a sacrificial context, and they may have been sacrificed because they were diseased, or they maybe in the process of incarceration were acquired TB. Okay, so what we found, I'm not going to get into the context, evidently. The strains that we see, and we retrieved, I think, 10 genomes here, and they date to this really tight time period just before contact. And what you'll notice is the lineages are deeper, so evolutionarily older, but chronologically younger than the strains from South America. So what this suggests, and we can calculate coalescence times of the different nodes, but what this suggests is really a pattern where we have some sort of early jump, and we know there were archaeological trade routes north, particularly between Ecuador and West Mexico by 400 BC or BCE. So they traveled north, and then there was probably, oh, I don't have that slide. Do I have that slide? Then there was probably a second jump that explains this variation, again, probably from seals and sea lions who were hunted and butchered. When we look specifically at the strains at Tlatelolco, we do see distinct lineages, but we also see what could be described maybe as something more of an outbreak type group. So we're very interested in applying modern genomic epidemiological methods to look at this further. But we have a couple of ideas. One, that maybe these weren't that transmissible. We don't see big strain replacement, but where we do see divergence, maybe this is because we know that the Spanish report that 65,000 to 100,000 people a day came to this market. And so the possibility of bringing, that's a great place for diseases, for pathogens to spread widely. Okay. What happened after European contact? We are looking at this, and we have some genomes and partial genomes from 3 contexts. One is Mexico City, one is in upstate New York, and one is in a community in Alaska. And there, everything we see is part of that red set of strains. So very quickly, we see replacement by the European lineages. They fall actually into different subgroups within the L4 lineages. And what's really interesting, so we don't have the subgroup. I don't have the subgroup yet for the Mexican one, but when we look at the Alaskan strains, they're in a really unusual or not common subgroup, but interestingly, very common or more common worldwide in Russia and East Asia. And of course, the Russians were the first to colonize Alaska and be in contact via the fur trade and the whale hunting. Yeah, that's a question mark. Okay, the other thing I should say is we don't have a ton of other data about TB from other places in the world. We do have an example from a former postdoc here at ASU. This is work she did during her dissertation from Bishop Peter Winstrup of Lund. We know exactly when he died, so we know how old this is. And you can see the coffin. I love the wood there. And this is a CT scan, and the arrows are pointing to these granulomas, these lungs, right? And calcifications. Oops, sorry. So when she did that, she, in her analysis, finds that when she dates these L4 lineages, they look like they really expanded during Roman times. The other thing that's quite interesting here, and I bring this up, so one of the things we'd like to do is, as we look at post-contact strains further, can we compare it to the types of strains we would see in Europe during the 15th, 16th, 17th centuries? And more recently, we have some selective scans of Europeans, particularly ancient Europeans. And what's interesting there, from Kerner et al., is you can see changes in allele frequencies for a susceptibility allele for TB. And it suggests that you see this sort of plunge, it sort of wanders about in frequency, but you see it sort of plunge as TB gets, reports of TB get a lot worse. And so one of the things we're interested in asking is whether we see, now this allele is not present in Native American populations, so, but we're interested in whether we see evidence for susceptibility or resistance to tuberculosis or other pathogens in American populations through time. So the last thing I'll end you with is actually just thinking about, when you think about disease and pathogens over time, you have to kind of think about what humans are up to and changes in human population size. And of course, during most of our history, prior to 10,000 years ago, we were hunter-gatherers with low densities across the landscape. And so it's really after the Neolithic, and then as settlement becomes denser and we live in close quarters with animals, this 4,000 to 6,000 years ago is really popping up at a time when a lot of these pathogens appear to be really hitting human populations hard. So I like to think about, when and where were different pathogens more likely pre and post Neolithic revolution. And, in the later parts, as the population densities increase, is really, I think, prime time. So, before I get to that, I should also say where the TB project is headed. We are now really intensively sampling in a certain part of Peru where we see the highest density of cases pre-contact, again, to try to apply these genomic epidemiological methods and see if they work over extended time periods. We've also started a collaboration with Eduardo Amarim and his group to think about these patterns of selection that might have affected the people as a result of these pathogens. Right. People in the lab wonderfully do a lot of work to move all this forward, and funding agencies obviously are helpful. But I really have to thank the descendant communities and national institutes who have agreed to participate in this research or have agreed to allow us to sample from these individuals. It is a destructive analysis, so we have to be mindful of that. Also, many wonderful collaborators at many places for these projects. So thank you. I went a little long.
I have one question, one pop culture question. Have you heard the song TB Blues by Jimmy Rogers? That'd be a good one. You could play it when you're giving a talk. He was a great music star in the 30s, and he died from TB. He's got a really poignant song about having TB. You can find it on YouTube. It's really interesting. My other question is, actually I have three questions. The second question is how do you diagnose in these ancient bodies the TB? Is it the granulomas or the spine? Like, how do you identify that this
Yeah.
person likely died from TB or?
Right. So most of the individuals that we have data from are not mummies, so we're not looking at granulomas. We are sampling, so TB leaves these diagnostic lesions in the bone. And so we're sampling near those lesions. So I collaborate with Jane Bykstrup, who's a paleopathologist here at ASU, as well as other paleopathologists, and that's really how we have sampled individuals.
Yeah, sure. Just ask this one. Give a playlist.
No, I don't have a song rack, but I'm sorry, I don't come with that. But so you mentioned at the beginning, like you were talking about, I guess, like the prep of these samples and the sort of sequencing approach that you guys use to get this data. And you mentioned a capture-based approach, and there's an assumption that you kind of know what you're looking for. And I guess, like, do you have ways in which, when you're unsure of what you're looking for, how you go about like sifting through all of that data?
Yeah. So when you shotgun sequence, if you're unsure what you're looking for, we will often use shotgun sequencing to go through and I don't know what I touched, but evidently, sorry, Suhail, the Grand Canyon. Right. So we've kind of gone about it in two ways. Early on, what we would do is we would use some of these repeat elements to do a qPCR to see if we could even just detect that TBDNA was there.
Right.
And then we would follow with a full-on capture. Now what we tend to do is a shotgun sequencing, and then there are a series of programs like Kraken and Malt and Hops that you can basically sift through all those data and bin it into different phylogenetic clusters, bins. And then you map to, we actually map to an ancestral TB genome. It's a fake TB genome because TB, if you know about the TB complex, there are all these deletions that we've basically put back in. Yep. And actually Iñaki Comas created this because it really makes it easier to map and align sequences even from modern genomes. So that's what we use there.
Gotcha.
Cool. So really cool stuff. Are you seeing more hypotheses in child skeletons or in adult skeletons?
So the distribution of lesions in children is different. So because children are growing, you will often see lesions in joints where in the epiphyseal plate area. And so often in legs and fingers. So the distribution of you'll still see POTS, occasionally POTS disease. I don't know if it ever gets, I don't think it's quite as common for children to get POTS. But Jane Bykstrup would be the one to ask that for sure. She would know. But they are affected a little differently. And one of my hypotheses is that we are testing some teeth. So there have been enormous numbers of assays in Europe, mostly for population history, where they've used DNA extracted from teeth because it's just generally a good place for DNA preservation. The other place people in Europe will commonly sample is in the petrous portion. Petrous portion is a really bad place to look for pathogens because there's not much blood flow. It's it's really dense. So it's great for hosts. It's not good for pathogens. So I thought maybe subadults, you might be, because it's a little bit more systemic, maybe, that you might be more likely to detect TB in teeth. We've had a few hits in our latest sample, but we won't know until we complete capture whether that's, if that's been successful. So hopefully have 16 to 19 genomes on their way.
It's me. Hi, Anne. I just had a question about your. So I don't usually work with ancient DNA. So for when you call variants, are there like certain tools that are ancient DNA aware? Because I'm assuming 1x coverage, if you put it in like the GATK, it'll not be good. So are there like
Oh, no, we'll still use GAT. But no, we try to get a higher coverage. So we'll look at library complexity and capture again, or do another sequencing run to try to get the numbers up. But yeah, so we run it through the eager pipeline, and then we will, part of that is it gets looked at through map damage, which trims off all the damagey bits. But at the end of all this, we end up with a VCF file and all that usual fun.
thank you.
We've got a question on Zoom, but I want to jump in and just take one question myself, which is, what's the common ancestor? Like, when did the two Mycobacterium lineages split? So the leprosy and tuberculosis.
Leprosy and TB. That's a good question. So M. lepra and M. lepromatosis probably split on the order of 10 million or so years ago. So the TB, I'm trying to think if there's been a recalculation of that, and I'm not thinking of one off the top of my head, but it would be millions of years ago.
So not like 6,000 years
Yeah,
ago.
not 6,000 years ago, no.
All right, sorry, Howard, I jumped your question. So Howard asked, would you expect genomic changes in pathogens following an outbreak as the number of susceptible hosts decreases? If so, would this be more akin to genetic drift or natural selection?
So during an outbreak, so there's a couple of things that we have to think about. One is, so there is variation within a host, but there often is a transmission skew in terms of what gets transmitted to the next host. And that is something that Susanna Sabine and Jeff Jensen and I have kind of thought about a little bit. And so we have those issues that are not super well studied. So then there's the whole debate about what happens when a new pathogen arrives in an immunologically naive population, right? How quickly is it going to spread? And what is the outbreak going to look like? And what is the population boom, really? And on top of that, you have something that was in a seal that has jumped into a human. And so that you expect at least some immune pressures to be different. And so there's a lot there to unpack, which is challenging. And you also have an organism looking at selective pressures challenging. So this is something that I should say I've also started a new collaboration with Lucy Van Dorp at UCL, and we're kind of trying to think about what might be the pressures that we see and how to detect those effectively. Some of these genomic epidemiological frameworks try to get at some of this. Those have mostly been used on outbreaks that last no more than a year or two. And so we're going to have a different time dimension, which may or may not make them terribly effective. So maybe.
Quick question. Yeah. All right, that's it. We ran a little bit over time. Let's thank Dr. Stone for a wonderful presentation.